Okay, I guess I will get started then. Hi everyone. So for those of you who don't know me, my name is Catherine Walsh. I am a programmer in the section of functional imaging methods here at the NIMH. And today I'm going to be talking to you. Come on in. Yes, sorry. Sorry, more people just walked in. Just so you guys missed that, my name is Catherine Walsh. I'm a programmer in the section of functional imaging methods, and today I'm going to be talking to you about studying brain behavior correlations with fMRI. So just to give you a bit of an overview on the talk of what I'm going to tell you about today. So first off, just want to define what brain behavior correlations are, so we're all on the same page, and talk a little bit about why we might want to study these, and then how to study them. So starting off with our first question. What are brain behavior correlations? So to break this down, I want to go through this definition. I'm going to break it down into the three words that make up this statement. So first about brain. This can be a lot of things. So it could be things from like activation, maybe in an ROI, in a network, could be functional connectivity, that could be at task, rest, any kinds of connectivity, structural measures, or other modalities, basically anything in the brain that you want to look at that's relevant for your research questions. Next is behavior. So this could be things like performance on a task. So things like accuracy, it could be reaction time, any measure that you might calculate from a task. This could be inside or outside the scanner. It could be self-report measures. So things like personality traits, psychiatric symptoms. So really just, this is pretty broad. And kind of finally correlations is some kind of statistical association or connection between the two things. And so this is going to be kind of a theme throughout the lecture that I'm going to be giving today, is that this is very much defined by your research questions. And there are some kind of overarching things to think about, and there isn't always a single answer for these questions. But yeah, we're going to get into that. So kind of moving on to our next question, why we might want to study brain behavior correlations. So before I go into actual brain behavior correlations, I want to kind of give you an idea of like what we might think about when like a traditional group level fMRI study. So for instance, in these kinds of analyses, we might have a population of people who we bring into we have them go in the scanner and do a task. And so maybe I show you a hard condition where I show you 3 faces and have you remember those for a short delay, and then ask you, give you a face and was this one you remember? Maybe there's an easy condition where you only see 1 face. I'll do some statistics and compare the easy and hard questions, and then get some brain regions that care about the difficulty of the task. So that's one great analysis. We learn a lot from those. There's been a wide variety of literature that tells that we can learn tons from. But there's more information that we can get. So for instance, if we put aside the group level analyses of condition, we could think about our people. There's more information. So people are doing this task, they're giving us behavior. And from there, we can kind of say that there's going to be variation. For here, the numbers don't really matter. So I'm just going to say that people who are colored in red might be doing better on this task. People who are colored more yellow are doing worse. We can rank the order of those and kind of get a sense about how people are doing on these tasks and the variation. Within each person, we can do a similar thing as we were doing before and compare easy and hard conditions, but we can do it for each person individually. That'll give us kind of similar things, and we can get brain maps for each person individually. And, we'll get a map for one person, and we can get a map for another, and maybe it looks kind of similar for the next person. And again, it's similar, but you can see that there's some kind of variation in those, and we can keep going and get them for everyone. And maybe some people are going to look a lot different. From those brain maps, we can then take some kind of measure. So like I said, that could be activation, it could be connectivity, whatever your question is. Do some kind of correlation between that and behavior. Activation in lateral prefrontal cortex is associated with better performance on this task. So still a new finding, just something slightly different than the other kinds of analyses that you might be looking at. Another reason we might care about these is looking at psychiatric symptoms in more of a dimensional rather than categorical way. So this underlying idea was when the NIH came up with the research domain criteria framework, also called RDoC. The goal of this framework was to develop, for research purposes, new ways of classifying mental disorders based on the dimensions of observable behavior and neurobiological measures. So just to give you an example of what that might mean, we might take groups of people who have some kind of DSM psychiatric diagnosis. So maybe something like major depression, dysthymia, and bipolar depression. And, if you go into therapy, you get a diagnosis, there might be, there's a different diagnoses, but they all share kind of underlying kinds of symptoms. So by putting them all into one big sample and trying to get more dimensional, continuous measures of symptoms, we can maybe better study them. And so this is a cool idea, but so what? Why is this useful? Thinking about psychiatric conditions in this brain behavior correlation allows us to better understand the underlying disease pathophysiology. It might help us track disease progression as kind of things move on, help us develop new and maybe more effective treatments, or for instance, understand who will respond to what treatments. So there is kind of all these different, these kind of useful in the psychiatric world ways as well. So I gave you an example of this visual working memory task and predicting cognition there, but that's not all that can be studied with this kind of framework. So for instance, one relatively recent review study looked across the literature and found about 50% of brain behavior correlations looked at cognition in general, which they then classified into kind of a more nuanced division. So some looked at things like intelligence, attention, but this really goes across any number of different kinds of varying degrees of cognition. As a couple of specific examples, work within the last decade has predicted, for instance, sustained attention from both task and rest, reading ability from connectivity in a reading-related functional connectivity network, things like fluid intelligence, and personality traits like openness and conscientiousness from resting state fMRI. So you might also notice that in this plot here, that even though about 50% are cognition, about another quarter of those are psychiatric symptoms. So other work has used these kinds of framework to try to understand what in the brain is, related to different different kinds of psychiatric symptoms. So for instance, these kinds of studies have tried to predict symptoms of autism, schizophrenia, Alzheimer's disease, ADHD, and depression. And that's just a small sampling of these. So taking a step back just to kind of review why we might care about brain behavior correlations. So things like basic science, where we get new insights into neural processes and a move towards brain-based psychiatry. And this might give us a better understanding about how diseases work, how they progress, and help us give more personalized targeted interventions, drugs, therapies, and so on and so forth. But wait, Peter started alluding to this in his controversies lecture last week. But there's some recent work that has asked whether most brain behavior correlations are even meaningful. This 2022 paper from Merrick et al. Took a look at some big open data sets, and in their paper, they suggested that to have reproducible brain behavior correlations, you need to have thousands of subjects. This finding was picked up by a number of different news outlets and generally led to some panic. So kind of led to these questions of, do we really need thousands of individuals to study these kinds of brain behavior correlations? And so just to give a brief overview of what they found, they took subsamples of varying sizes from the ABCD study, which is a big study of adolescent brain cognitive development, which has thousands of subjects. They calculated resting state functional connectivity and then tried to relate that connectivity in the default mode network to two behavioral measures, the overall cognitive ability and different measures of psychopathology. And what these panels are showing here is that this across the different sample sizes, the correlation between the resting state connectivity and the behavioral phenotype, which is here on the y-axis, varies a ton with smaller sample sizes. And eventually, when you get up to kind of the thousands of subject level, it starts to converge to a stable mean. They took this further and showed that you can pull specific samples of 25 subjects, so things that we might see as a sample size in fMRI studies, and you can get drastically different effects just due to sampling. And so you can see here for both of these, we have a light green line that shows this really strong negative correlation and this darker green line that shows a really strong positive correlation. And so that just due to sampling variability, you can really get drastically different results, even if you can see this dotted line is the correlation in the whole subject with almost 4,000 participants. There is no relationship. So this really underscores that there are widely varying effects at small sample sizes, even of which can produce completely opposite results. So this was scary. But that being said, there are ways to do this in a robust way. So I really just kind of blew through some introduction, but I want to finish kind of spend most of my talk talking about how we can robustly study brain behavior correlations. And again, as I alluded to this before, a lot of this is going to kind of come down to your specific question. There are different ways to do things, and there's no one right answer for a lot of these. So how can we robustly study brain behavior correlations? The first one obviously is sample size. If you get a bigger sample size, it is easier to study things more robustly. And there are obviously benefits to that. Correlations get more reliable. This is what the Merrick et al. Paper showed. Another benefit of a larger sample size is also that you can get a wider distribution of phenotypes. So for instance, if something varies a lot, you can get that variability better in a larger sample size. It also allows us to use more robust statistical and machine learning methods like cross-validation, and I'm going to tell you what that is shortly. However, despite these benefits, there also can be problems with using a larger sample size, oftentimes on the practical side. It can be expensive. If you're paying for your subjects, it can be a lot of money per scan time. It also is time consuming. If you are trying to get 100 people in or 1,000 people in for 2 hours a pop, that's 2,000 hours of man time. That's a lot of work. And also, if you're working in a psychiatric patient, it can be hard to recruit patients. So sometimes if you have a smaller psychiatric population, it's just not feasible to pull in 2,000 subjects. So what if I can't recruit thousands of subjects? What can I do? Am I just out of luck? No, there are things you can do to kind of mitigate these issues. So one thing you can do is to consider getting more scan time with fewer subjects. There's been some recent work that has kind of looked at the balance between having more scan time versus more subjects. What we can see here is this plot from this preprint where they show they basically vary the amount of number of participants versus the number of scan time per participant, and show what the correlation is with various behavioral phenotypes. And what you can see here is that there are these lines which are that same correlation, which you can kind of get you kind of look at that trade-off of, it is possible to get a similar kind of connection between brain and behavior, depending on various different subjects versus scan time. An earlier study showed the similar effect with fingerprinting. This is an analytic technique, which is slightly different from brain behavior correlations, but essentially is, given some sets of data, can I tell your brain scan from my brain scan? And they showed that you are able to do this with better accuracy as you get more scan time. Finally, having more scan time allows you to do kind of fancy individual parcellations, which basically, as opposed to taking group level parcellations, looking at kind of where there might be activity and finding places of individual variation, which may improve prediction accuracy relative to other group defined parcellations. So you can kind of see that this MS-HBM, you can see that this prediction accuracy is higher than these other kind of standard parcellations. Another thing that you could consider is use what measure you're using. And I'm going to talk about different measures that you could use. The first one is task versus rest. So many studies use resting state fMRI to predict behavior, and there's a lot of good reasons for that. It's relatively reliable across sessions. It's kind of trait-like. We think that it reflects functional networks that might be a backdrop to anything that happens during tasks. It's relatively easy to measure. There's no task to learn. It requires relatively little scan time to do overall. And many of the big open data sets use it. So if you have these big publicly available data sets, they often include REST. So you can kind of check the number of subjects box. But is it really best for prediction? And there is kind of burgeoning work that's suggesting that maybe not. So there's been some recent work that has looked at 2 independent data sets, one from the Human Connectome Project and one from the Penn Neuropsych Consortium, where they tried to predict fluid intelligence from a number of different scan tasks in addition to REST. And they found that connectivity from these scan tasks, so all of these, basically, these are the various tasks in the Human Connectome Project, were better able to predict intelligence than REST, these last two bars here. And a similar result was found in a separate independent data set. We can kind of think about this task versus rest question as like a stress test. So Emily Finn, who's done a lot of work on this, has proposed that we think about a good analogy is like a cardiac stress test. So if there's something, you think there's something going on with your heart, you're not going to try to look for something going weird if you're sitting on the couch. You put someone on a treadmill to kind of see what's going on. So that's one way to think about that task could be kind of pushing these individual differences to make better prediction. That being said, you could also use both. There's nothing to say that you can't use more complex analytic techniques that allow you to integrate that. The review paper that I was showing you earlier did look at various papers that either try to integrate across modalities, so including task and rest, or task and structure, or rest and structure, any combination, and found that those tended to do better than prediction algorithms that just used one kind of aspect of function. It's possible that integrating across neuroimaging features can improve prediction performance and leverage unique aspects of brain structure and function to better characterize behavioral traits. So those are kind of things you might think about in terms of the measures from the scanner. You also might want to think about the measure you use on the behavioral side. So things like self-report versus a cognitive task. So just to kind of show you what I mean for that, take, for example, face blindness, so prosopagnosia. If you want to try to see how well people, or whether people have this condition, one thing you could do is ask them, use self-report measures. So for instance, you could use the 20-item prosopagnosia index, where people just read these questions and kind of endorse how much that applies to them. You know, my face recognition is worse than most people. I have always had a bad memory. So just asking your participant. Another way you could measure this is by having them do a cognitive task. So for instance, the Cambridge face memory test. So where in this task, you have people who see a face, try to memorize it, and then are given a 2 alternative force choice as to, was this one of the ones you memorized? And then by that way, you can experimentally test how good is their face memory. And again, like before, there are pros and cons to both of these. So for instance, for self-report measures, they're easy to administer. They might be more relevant for psychiatric symptoms. It might be harder to develop a task to test your psychiatric symptoms. And they're potentially more stable within an individual. However, these do have the potential for bias a couple different ways. If you have a person who knows they're coming in to test their face memory, they might be more biased to say, oh yes, I have really bad memory, just because they know that that's what you're looking for. And they do also rely on a person's ability to introspect. So are they aware of how good or bad they are at something? In terms of the data from a cognitive task, there might be less potential for these kinds of biases. You could do these in the scanner. So if you want to directly relate the brain activity to what you're doing on a task, you can do that a little bit easier with a cognitive task, and they might be easier to predict. So one recent study using the Human Connectome project looked at these kind of various prediction methods and across a number of different behavioral measures from the Human Connectome project, and found that task performance tended to be better predicted than these self-reported measures, so these kind of questionnaire measures. Cognitive tasks, however, do have some cons. They might be less relevant for biomarkers. So if we're looking for some kind of generalizable, marker of a psychiatric issue, it's harder to say this one task is relevant for all psychiatric stuff. And kind of because they are potentially less generalizable. A task is trying to measure a construct, but there are, little differences across any given task that are idiosyncratic. So we've talked a little bit about the kind of behavioral tasks you might use, and now I want to think a little bit about what exactly kind of task measures are comprised of, so we can help to try to decide what task is best. So if we take a task, be it a cognitive task or self-report measure, we can try to break down the sources of variances to try to understand someone's score. So on one hand, we're going to have true differences across individuals. Maybe Tyler is just better at memorizing faces than I am. There's some difference that is kind of stable. Tyler is saying no, but I don't know if that's true. But yeah, so there's true difference across individuals. It's just going to happen across time. However, we're also going to have things that we call within subject variance. So these are things like metabolic chains, physiological state, arousal, attention. So if I had my coffee this morning, maybe I'm going to be better at this task than I would be if I didn't, or if I didn't sleep well. These could also be things like experimental condition. Is this an easier task? Did I give you a distractor during the task, or is it, give you 3 faces versus 1 face? Then to fill out the pie chart, we also have things, error, measurement, noise, randomness, so on and so forth. And we can think about these kind of breakdown of between versus within subject variance for any number of tasks, and tasks have different ratios. One recent study looked at the various measures from the Human Connectome Project and tried to classify them in terms of this ratio and found a wide range across the tasks. So we could have things on this kind of top lefty area, which are measures that vary a lot between individuals, but are relatively consistent. So if I ask you to do this task over and over, I'm going to get similar scores, but I'm still going to be very different from someone else, versus these measures more on the bottom right, which have a lot of within subject variance, but not a lot of between subject variance. So as a whole, we're all going to look pretty similar, but I might be really different day to day from myself. And so the one thing that I want to highlight about this kind of question about between versus within subject variance is that these can both be interesting targets of prediction, but they're just asking different questions. So when we think about brain behavior correlations, a lot of the time we are thinking about between subject questions. Understanding scores in a working memory task or overall symptom scores and things like that. But within subject variance can be just as important. These could be things like fluctuations in psychiatric symptoms across a lifespan, understanding how some sort of cognitive task or cognitive process develops over time. And these are just as important, they're just different. And so kind of with that in mind, different questions necessitate different kind of tasks. And I'm going to give you a little bit of hand wavy math and statistics to show you why. So no equations, but just bear with me here. So we can first think about within subject effects. So these are things like the group and condition differences that we see in kind of classic fMRI experience. These often look at t-tests to look for significant differences, and without using any math here really, this is just looking at the difference in condition over the error. Thinking about our pie chart again from before, we could break down that error into the variance in individuals and random error. So then kind of moving towards on the other side, we could have our between subject effects. So these are things like brain behavior correlations. One way we look at this is often the inter-class correlation. I'm not going to tell you I don't do on the math of what that is, but essentially that's looking at the variance between the individual given the error and the measure. So if we want to maximize our chance of finding a difference in conditions in a within study design, so that's these types of questions here, one way we can do this is by minimizing the variance between individuals. And so that way with arithmetic, that makes the numerator bigger and that makes our t-value bigger. Great, wonderful. But say we take that same task where we've tried to maximize the variance between individuals or minimize the variance between individuals, excuse me. That's going to minimize the numerator in our other between subject measures and therefore make the thing that we care about in terms of between subject effects smaller as well. The long and short of this hand wavy math is that tasks that are optimized for within subjects effects might be poorly optimized for between subjects effects. So kind of thinking about, if I know that the Stroop effect is really, really strong and I see it in everybody, that might not be the best type of task to use for these between subject correlation, these brain behavior designs. And I've got a citation here. This will be online if you are curious about the math and want to see a little bit more of the stats behind that. So one other thing to think about in terms of optimizing sources of variance, you can also think about that in terms of what population you're sampling from. Specifically, if you're looking for a specific behavioral phenotype or a psychiatric profile. So that going back to that meta-analysis, they showed that studies that were trying to predict things from patients tended to do better than those that predicted from just healthy controls. And there are a lot of caveats to that, and I'm not going to get into that too much, but just as kind of a thought experiment, consider that you're trying to find some sort of psychiatric phenotype in a sample of healthy volunteers. There might not be just enough variance in those kind of the healthy volunteers. They might just not be showing those psychiatric symptoms. So your model won't be able to pick up on that variance and you'll get really, really crappy prediction. So yeah, so those are kind of big pictures in the terms of the measures that you might use. Another way you can think about is improving your data quality. So this slide, which I took from Dan Hanworker's lecture a few weeks back, we can see that there is a relationship between the number of subjects down here on the x-axis, the effect size. So this blue is kind of a base effect size and the statistical power we have, so the ability to see an effect. And you can see that if you have a bigger effect size, you're able to see an effect more clearly with fewer subjects. So while these slides were designed to think about group differences, the same logic follows for these kind of between subject analyses. If you have a clearer effect size, you'll need fewer subjects to see a clearer relationship between brain behavior. And so that's kind of all in just theoretical. Let me give you a few practical ways that you can improve your data quality. So the first that I'm going to talk about is briefly is multi-echo. Again, the slide from Dan. One way is thinking about your acquisition parameters and pre-processing. So here specifically, Dan shows a few different pre-processing pipelines into regions that are doing visual processing. And the big picture is that if you use multi-echo acquisition and kind of these state-of-the-art pre-processing methods, you can get a drastic improvement in your effect size. I'm not going to go into a lot of detail on this. If you're curious and you missed the lecture, go back and watch it online. But yeah, there are things you can do to improve your data quality. Another thing you could do is try to decrease head motion. What I'm showing you here is a paper that looked at, again, the Human Connectome project, that head motion is significantly correlated with a number of different behavioral measures that they collected in this data set. We also know that we do tend to see greater head motion in certain populations. So kids, older adults, psychiatric populations. If your behavioral outcome is correlated with head motion, it's going to be harder to predict. I'll also note that there does tend to be less head motion in tasks relative to rest. So that's another point in favor in doing a task in the scanner versus rest. Another way to think about improving your data quality is by maximizing reliability. So I'm going to go into a little more detail here about this. So a key assumption of brain behavior correlations is that we are measuring stable trait-like things. That is to say, the things that we are measuring are reliable. And let me give you a kind of brief overview of one way of thinking about that. So say I have you do a task. It has a number of trials. You get either correct or incorrect on each of them. And on the first trial, you get it right. Second trial, you're doing just as well. Awesome. Third trial, you kind of space out a bit, you get it wrong. Fourth trial, fifth trial, you're doing well. Sixth trial, you mess up. Overall, you got an accuracy of 0.66. So great. What if I try to split these into two subsets? Maybe we split out the even and odd trials. If we calculate the accuracy on both of those samples of trials, we can see that it's the same as our overall accuracy. We could shuffle the trials again and we can do the same and get similar accuracies. Try it again, do the same thing. We're getting the same accuracies. If a task is reliable overall, we'll mostly get the same accuracy across any number of trial splits. And what that's suggesting is that we have high split half reliabilities. That is to say that the task you're using is internally consistent, that it's really measuring the same construct all the way through. And there are other ways you can measure this, but this is just kind of one way. One question, though, about the split half reliability is whether all tasks show acceptable levels of split half reliability. And if not, how can we figure out which tasks do? So this is some work that I collaborated on, and we set out to test exactly this. So we took a battery of behavioral tests and split the trials into different subsamples. We correlated the scores across these subsamples, repeated a number of times, and to get a distribution of the correlation of scores across subsamples. We took the mean of that distribution and repeated that across different number of trials. So the first time we might have taken 2 trials, correlated the score, go on. Then 5 trials, correlated the scores, 25, 100, so on and so forth. So what we wanted to test is whether there's this split half reliability stabilized at any point to see if, I could say it took this many trials and then I'm stable across sample size. What we showed is that for some tasks, so for instance, that Cambridge-based memory task that I alluded to before, it does stabilize. So you can see that kind of around somewhere in this kind of 100 trial-ish range, we kind of start plateauing and that there is strongly reliable no matter how many trials of this task that we use. We did this for 12 different tasks, which I'm not going to show here, but that was not the case for every task we took. So for instance, we also had people do this emotion labeling task where they saw a face that was making an emotion and had to say which of the 6 core emotions was this. And it really it never plateaued. It required many, many, many trials, like thousands of trials to even start to plateau. So this is not always the case for every trial. And the number of trial it takes to reach an acceptable level of reliability varies across task. And importantly, that doesn't always correlate to the number of trials that are included in a single form of like a given task. So if you take a standardized task, that's not always the number of trials it takes to be reliable. Okay, so what? Some tasks are less reliable than others. What does that mean for what I'm trying to do? So to show that, we took two tasks that we knew that should be relatively well correlated. So for instance, a car memory task and a task where you saw a car and then saw it for 500 milliseconds and then had to say which, match it to another. We subsampled trials to get them to be of various levels of reliability, took the scores from each task and correlated them. So what I'm showing here is the results of that. And so basically what this is showing is that at varying different reliabilities, at lower reliabilities, there's a lower correlation. So when tasks are less reliable, they show attenuated correlations, even when they are truly related. So even when we know they are related, you aren't going to see that as well if you're using less reliable tasks. And now I'm showing this for two behavioral tasks, but mathematically this is the same for brain behavior, brain, any kind of correlations that matter. This is the same. So that was split half reliability, but there are other kinds of reliability we might care about. So for instance, we might try to relate some kind of brain measure from an MRI scan to some kind of behavioral measure. And on that day, might be a correlation of 0.4. If that relationship is reliable, we might expect that time passes, could be hours, could be days, could be months, could be years, it doesn't matter what the time is. We could do it again on a second session, repeat the process and get a similar correlation. This kind of reliability is called test reliability. If you kind of repeat a measure, you'll have a similar relationship across time. And so what I showed you in the last analysis, I showed you is that you need to have enough trials to have a reliable task. You need to have a reliable task. But that brings up another question. So if there's not enough trials in a single task, can I just do two? I can just do two forms of it. Great, easy. But do those have to be, can I do those on the same day? Can I just say do form 1, then form 2 right away? Or do I have to have split time? Oftentimes when we're doing these big studies, we have people come back. Does time matter? So to test this, we had 3 batches of non-overlapping participants, which did 2 versions of the Cambridge-based memory test with 3 different kind of timing. The first were on the same day, so they just did Form A, then Form B. The second was a few days apart, so they might have done Form A on Monday, Form B on Thursday. And the next was 7 to 8 months apart. And so theoretically, we said that if these sessions are equivalent, i.e. it doesn't matter when they did it, test-retest reliability should be the same as split half reliability. So just to kind of put that in other words, we did the same analysis. We sampled trials across time, and we kind of did this in 2 different ways. The first was by pulling trials, just agnostic of time. Doesn't matter whether it was, day 1 or day 2. I just took some trials. I correlated the samples. In the second way, we kept in mind the timing of them. So when I pulled my first sample, they were always from day 1, and I pulled my second sample, they were always from day 2. And so we can make those same plots. Here, the light colors reflect the test-retest reliability, where the forms are kept separate, and the darker colors are split halves reliability, where the forms are pooled across the days. And what this showed is that pooling data across sessions increases the reliability the reliability of measures. So suggesting that even within it, so that having multiple days and more time in between, you might kind of help cancel out some of those within subject variants that I'm really tired on this day and I'm awake on this day, and you kind of help get a better measurement of these true trait-like differences. And yeah, so just in a quick plug, we did create a web app to help explore the reliability from my pilot sample. So we did some cool derivations of the map to find the relationship of number of trials you need to have a reliable task. We made this in a nice usable web app formula, so you can take some pilot data and figure out and make sure you're doing tasks that are good for individual differences work. Okay, that was a lot. And these will be online, so don't worry about writing down the link. And so the last thing I want to talk about, just changing directions a little bit, I've talked a lot about ways that you can think about optimizing your design and your data acquisition. And so now I'm going to talk about some analysis techniques to make sure that you're not biasing your results. There will be a full lecture on these types of methods later, so I'm going to kind of give an overview, but I did want to give a quick example. So the first thing I'm going to talk about is overfitting and why it's bad. So imagine you have some kind of variable X and another variable Y. Maybe that's activation and ROI and a behavior. Maybe that's two ROIs. Doesn't really matter. And we can see here that the way I made this distribution, they are related. There is a linear relationship. I can take my full sample and I can do some statistical modeling, and I can fit a model to it and get a really, really specific model that will predict my data really, really well. But if I try to then think about like what happens if I take a new sample of people who from the same underlying distribution of people, and I try to use the same model to predict it, it's not going to work as well. If we try to use the same model we used before, there's going to be a lot more error, and a more simple model like the linear fit will just serve us a lot better. This all is to say that creating a model using our entire sample can lead to fitting sample-specific characteristics, so things like noise, and lead you to think you're doing better at predicting than you actually are. So how do we fix this? One way is cross-validation. I'm going to give you a toy example of what this might look like. So say we have our sample data. We could split this into a training set and a test set. We will train our machine learning model in our training set, and then we'll get predicted values from that. Doesn't really matter for the moment. But then we can take that model and apply it to our test set. From there, we'll get a predicted value from our test set. And keep in mind here that this test set, the model never touches this. It never sees it. It's completely new data for this model. We can compare our actual value to a predicted value and then see how close they are. And that can be our measure of, that can use as a measure as how well our model is doing. To make sure that we're really generalizing our data well, we'll often use what we call k-fold cross-validation. What this does is to iteratively break our data up into non-overlapping train and test sets and repeat this whole process. So just let me show you what that means. That was a lot of words. So if I have my data set, and again, I say I have 6 people, first I'm going to break my data into a training set and a test set. I'll train my model, I'll make my model in my training set, I'll apply it to my test set, and I'll get some score. Then I'll do the whole process again, but I will use a different test set this time, different training set this time, get another score. Do it again, third round, different test set, different train set. And I can do this for as many times as I want. I can break this down into as granular as I want, even down to just holding one subject out. I could do this in fewer samples. It really just depends on how big your sample set and kind of how you're doing, like specific parts of your modeling. But I can get my scores across my folds and average those scores from across the rounds to kind of get a sense overall as to how my model is doing. So I want to just show you some actual data to show how important this is. So a recent study took the data from the Human Connectome projects and tried to predict behavioral phenotype with and without using cross-validation. So what they did is they trained a model in samples, various different sample size. That's the color here. They did it in a discovery sample, so in one kind of subset of data, and then ran that same model in a new discovery set, a new non-overlapping set. When they didn't use cross-validation, they just trained a model and those kind of went with it. You can see that they did a lot better they did a lot better than you'd expect. That the sample is biased to be higher in the replication sample, suggesting they're overfitting. When they use cross-validation instead, you can see that it's more, these values are a lot more on this kind of identity line here. There's more similar across the discovery and replication sample. Shown another way, we can look at the second figure. This is the x-axis here is sample size that got cut off. I apologize. But they can kind of look at the difference between the correlation between the discovery and the replication sample when they had without cross-validation in the red and with cross-validation in the blue. And when they don't use cross-validation, their correlations were drastically inflated, and that bias was pretty much gone when they used cross-validation. So Yes, so the long and short of this is that without cross-validation, effects are overestimated in the discovery sample, and that it helps unbias the effect estimates. And so cross-validation leverages held out portions of your data to see whether your models generalize, but a more stringent test of how well these models generalize is to use an entirely independent sample. So for instance, in a study from Monica Rosenberg, they used task and rest data to predict scores on a sustained attention task, and they were able to do really well. Their model let them see which nodes of functional connectivity were particularly relevant, so they could use this to define a network that they called sustained attention network. They then took this model and applied it to an entirely new sample of, they applied it to a new sample of kiddos with ADHD and used this model to try to predict their ADHD scores. And they were still successfully able to do that. So the fact that the model they developed in healthy controls was able to predict ADHD scores in a new sample was a great test of how useful and valid this model was. Yeah, so kind of wrapping it all up, I know that was a lot of content. I'm happy to go back and answer questions. But our question was, what are brain behavior correlations? It's the statistical association between some sort of brain measure and some sort of behavioral measure. And why should we care about them? They give us new insights into nuances behind neural processes and support the move towards brain-based psychiatry. And finally, how can we robustly study brain behavior correlations? We could increase your sample size and/or your scan duration. We could think critically about what measures you're going to collect, both on the brain side and the behavior side. Improve your data quality, again, of both brain and behavior. And use appropriate statistical and machine learning methods like cross-validation and generalization into an independent sample. So lots of people helped with this projects. Thanks to Dan for giving me slides and for collaborators for collecting and analyzing this data that I've presented today. So yeah. I'm happy to answer questions. I know that was kind of a whirlwind.